Packaging Phage-Lambda DNA in vitro. If we are going to synthesize recombinant DNA molecules by using lambda genome, so this is also very important steps that we can manage in vitro packaging of the recombinant DNA molecules. Naturally, if we use a lambda genome which has not been modified and it is a wild type, and if we use it to infect host organisms like the E. coli, then the plaques may be up to 10⁶ plaques per microgram of lambda genome it is there. So the efficiency of plaquing it is more or less optimum if we are going to use unmodified lambda genome. On the other hand, if we use modified derivatives, then the frequency may be reduced. For example, the plaque formation may be up to 10⁻³ to 10⁻⁴ plaques per microgram of vector DNA. तो इसकी क्या वजह है कि ये जो प्लेक फॉर्मेशन की एफिशिएंसी है ये कम हो गई है? तो इसमें आपने एक डायग्राम में देखा होगा कि जब लैम्डा जीनोम के दो फ्रेगमेंट्स दैट आर एसेंशियल वन वो लाइगेट करते हैं फॉरेन डीएनए से, तो डिफरेंट पॉसिबिलिटीज़ हो सकती हैं, जिसमें एक पॉसिबिलिटी ये है कि डीएनए के जो रिकॉम्बिनेंट फ्रेगमेंट्स बनते हैं वो छोटे होते हैं, दैट आर नॉट ऑफ एप्रोप्रिएट साइज, तो उसकी वजह से फेज की वाइबिलिटी जो है वो रिड्यूस हो जाती है. चूँकि फेज की वाइबिलिटी रिड्यूस होगी तो उससे जो प्लेक फॉर्मेशन होगी वो भी कम हो जाएगी. सिमिलरली, another reason for low frequency of plaque formation when we are going to use modified lambda phages is that the DNA fragments they are produced are very large, that are their size may be more than 54,000 base pairs that cannot be packaged into the phage head. So in this situation, non-viable phage particles they are produced that can result in the low frequency of the plaque formation. On the other hand, if we use optimum length of the recombinant DNA molecule, that is between 40,000 base pairs to 53,000 base pairs, then we can get viable phage particles. And by using modified lambda phages, the examples that I have already quoted, the frequency again can be increased up to 10⁶ plaques per microgram of lambda DNA or the vector DNA. So in this way, the number of plaques formation, it may be reduced or even optimum if we are going to use modified lambda phages. This diagram, it indicates the in vitro packaging of the lambda DNA. In the first step, the requirement, it is the head precursor, and the head precursors, they are being synthesized by the gene E. And in the next step, the concatemeric DNA, it is inserted into the head. As I told you that concatemeric DNA, it is that type of DNA that contain multiple copies of the same DNA. For example, if lambda genome is there, so number of copies of lambda genomes are there that are separated by the cos or cohesive sites. So because it contains multiple copies of the same DNA, so such type of DNA, it is called as concatemeric DNA. And during the step of packaging, product like the gene A, it is having endonucleolytic activity. It will cleave the DNA at cos site and convert it into monomeric form. And this monomeric form, then it will be packaged into the phage head. And then the product of gene D, they are also included during the encapsulation process. And finally, some assembly proteins that included the products of gene W or F2 plus the tail components, they will ultimately result in the production of mature phage particles. So in this way we can get the phage particles that contain our recombinant DNA molecule. But under natural conditions, the situation may be that this lambda genome, it is effectively packaged in the form of concatemeric DNA. But in vitro, we can also provide the monomeric DNA molecule if high concentration of vital proteins like the phage packaging proteins that included the head or tails are also present. So in vitro packaging can also be effective when we supply the phage particles with monomeric DNA.